you, Luisa. Thank you, everyone. So I'm presenting today on the ERGA pilot, I should say, on behalf of the project coordinators, which is me, Anne McCartney, and Alicia Mouton, and of course also on behalf of all the many participants in the pilot project, as we'll see in a moment. Just to say that essentially, now probably ERGA is one of the largest, if not the largest, biodiversity genomics community in the world. But at the time that ERGA was starting, that wasn't the case. Now it was still growing up, and basically, there was this idea of establishing a first project that could showcase the importance and the power of this approach. This was before BG, before everything happened. But now, gladly, we're finalizing this pilot project. So I'll talk a little bit about this in a moment. Before we even start that, though, I would like to acknowledge that again, this is really a testament to this terrific effort from so many people across so many different countries. I don't think this is unfortunately inclusive, but it really testifies to how this was a collective effort from Europe as a whole. So we're talking about people, we're talking about institutes, we're talking about companies that were involved. A big thank you, a big shout out to all the people that made this possible. I would say the first main outcome of this is the ERGA pilot preprint. So you can look it up now. If you Google European Reference Genomatous piloting a decentralized approach to equitable biodiversity genomics, you'll find all the results of the pilot. And of course, again, this was a contribution from many people. I hope we managed to list everybody. This is going to be hopefully published soon as well, and a journal, so stay tuned. All right, so what was the goal? Now, if you go back to the beginning, what was the goal? The goal was to really build and test a distributed infrastructure for reference genome generation, accessible, equitable, and that can respond to diverse needs of all biodiversity researchers, regardless of where they are located and their social political reality. So we are talking about taking care of all the steps of the way, from sampling to DNA extraction, DNA sequencing, data, genome analysis, and so forth. And this is what the final sort of workflow in the pilot looked like, from establishing genome teams, basically grouping together researchers with different expertise that could contribute to a specific project. Then we had, sampling procedures, sampling and metadata acquisition. That's something like, for instance, Tom discussed briefly before. Of course, then there was the wet lab, and then all the bioinformatic analysis. So let's dive a little bit more into this. In terms of sampling, first of all, we had to ask ourselves what species were most important, and there were a few people involved in this effort to really come up with criteria that could address this question. What are we going to we don't have a limited resources. What we're going to prioritize, and this was, I think, a preliminary effort, but that was really instrumental also for the next phases of the ERGA project. In the end, we came up with 98 species that were proposed, and there were samples available and so forth. Here is how they look like. They're all listed in GOAT. For those who are not familiar with GOAT, is this database, online database, where you can actually find the status of ongoing reference genomes projects. And in terms of genome team establishment, we really had a good variety of people from different countries and different places. You can see a schematic representation here, and there was also some international components to that. So in terms of pre-sampling procedures, this was really important, and was really a significant effort to try to come up with the standards, SOPs and other strategies to that end. So for instance, here you can see there was a sample code of practice that was put together, led by Anne, Astrid, and others. And essentially, again, this was an effort to try to standardize this sampling process across many different countries and institutions and taxa as well. So, of course, the sort of associated task here is to be able to associate the samples with the actual metadata. And to this end, COPO, this software from the Arlen Institute, was sort of selected as the strategy for this. COPO allows you to essentially, this was already described by Tom, so I'm not going to spend too much time on this, but basically sort of a sample manifest, okay, that can be potentially updated if there are new metadata fields and entries that you want to collect. But the idea is that you can collect all the metadata associated with the specific samples in a standardized way. You can have ontologists to validate the actual entries, and then this is your sort of finished product that you can use to submit your data to the public domain. The data extraction and sort of the sequencing happen in many different places. It's really, as you can see here, a continental effort, from sampling localities in black to biobank specimens, collection of vouchers. Now, in a lot of the, I think I have another slide on this, but basically a lot of the samples that we that ended up in the pilot were actually vouchered samples. And then, of course, library preparation hubs. None of this could have been possible without the many sequencing centers across Europe that would contribute with sequencing. And then, they could be distinct. Sometimes the library prep was done in the same place as the sequencing. Some other times it was separate. So you can see it's a very collective effort. We are in terms of sequencing, as I mentioned briefly before, this was made possible also by the contribution of companies. So I think we're really thankful to PacBio, ONT, Phase, Genomics, Arima, Dovetail, to contribute this effort that, again, it didn't have any funding at the time. So it was really possible thanks to these contributions, and of course also by a lot of involvement and direct contribution by the specific institutions that were involved in the sequencing process. In terms of genome assembly, this is something that, again, Tom already touched base upon, but basically pipelines were either adopted with some modifications or developed for both the assembly, the QC, and taking making use of the expertise of the Sanger manual curation team. Essentially, all the genomes in the pilot were subjected to manual curation by the Sanger, and will be, because they're not all done yet. And indeed, these are the assemblies so far. So here it's a non-final draft. You can see the white dots here. Pre-curation, like a few of them are in pre-curation, and some others are in curation. So there is, and this is not the 100 species. Now we're still sequencing or even sampling in some cases some of the species. But it's starting to be a pretty good amount. The available data sets were already analyzed. Now, this is something that if you want to know more about, you can go and take a look at the manuscript of the paper. Basically, you can see now that that all the samples were analyzed in terms of quality metrics, like scaffold N50, QV, completeness, in terms of k-mers, in terms of genes, and so forth. In terms of challenges, these highlighted a lot of challenges that some people are familiar with. Others were more subtle. For instance, in terms of the fact that for many organisms, we're not usually able to sample a single species or a single genome, but we have a lot of contaminants. These are two examples of the Cladonia norvegica. And you can see now what says here that lichen genomics is metagenomics, essentially. Or these other species of Platyhelminthe on the right, with parasites, essentially. So other problems that are, because of the breadth in terms of species, considering all eukaryotes, include, for instance, problem with nanopore sequencing, jamming the nanopores, or the fact that some samples are too small, so it's really difficult to actually get the sequencing data out of such a small samples. And as a sort of general findings in terms of things that can be potentially improved as we transition from the pilot to the actual project now, is in terms of capacity distribution. So of course, well, you can see it from the map now that there are some countries that don't really have a lot of capacity, particularly in Eastern Europe. This is particularly true when it comes to sequencing and diverse participation. Now you can see there is a bit of a gender imbalance here. A technical challenge is definitely that of sample shipment, like being able to ship samples across countries. These are 3 examples where this was particularly difficult. Then sample quality, it's really important to stress that reference genome production requires very high quality samples. This is not something that is so clear. In the time at the time of sort of Illumina sequencing, it was much easier, but now you need much better DNA as an input, and allowing the sample collectors who are really on the field to understand that this is a critical need. Otherwise, everything is wasted. It's not as easy. So as you can see, it's for many people that was the first time they were sampling, and that was difficult to explain. I mentioned before that vouchering, which is a critical need to make sure that there is a physical specimen associated with the actual sample, is something that was carried out in most cases, which is really good. Final word. Again, I mentioned that this wasn't something funded by, for instance, the European Union at the time. So it's really thanks to all the people who contributed that it was made possible. These are some numbers that Anne put together. So basically, for a genome, there is this estimate of about $26,000. Now, if you include everything, from collection to biobanking, coordination, and so forth. So if you sort of make the math and multiply this by the number of species that are going through the pilot, we can estimate this to be above $2 million worth of investment. And again, thanks also to the companies that supported us. So I think this pilot has a lot of ramifications. These are a few examples. These are projects that are really direct spin-offs in a way. Now, one is, of course, the Biodiversity Genomics Europe project that was funded with 20 million euros by the Horizon Europe. And another one that is very significant is the Atlas C, for specifically to generate reference genomes for species in France, and received a significant funding from the French government. And of course, a lot of nodes, national nodes, like from Sweden, Germany, Greece, Norway, etc., some of which have already established sources of centralized funding. I think another sort of inspiration, at least, was for Australia. There is a project that uses almost the same acronym as ERGA. With that, I'm going to finish. So again, thank you to all the participants in the pilot. Thank you so much. It was really fun to work together. And of course, we're not done yet, but hopefully we'll be able to again publish the first manuscript soon. Thank you all.